Artificial fertilization reveals key protein in sperm-egg fusion. Artificial fertilization reveals key protein in sperm-egg fusion. You're listening to Biotechniques Reading, presented by Hakeem Ali-Bocas Alexander on the Wisdom app for World Reading Club in association with UniquilibriuM. Today's reading focus is from Biotechniques.com. Biotechniques, the International Journal of Life Science Methods. The article is titled, Artificial Fertilization Reveals Key Protein in Sperm-Egg Fusion, 14th of September 2022, written by Aisha Al-Janabi, assistant editor. It's in the category of diagnostics and preclinical news. This is a cold reading for World Reading Club. Biotechniques continues. An international team of researchers has discovered a fundamental protein in human fertilization called MAIA, which could lead to an improved understanding of infertility. For fertilization to be successful, the sperm and oocyte, an immature egg, need to recognize and bind to each other before fusing together. A study led by the Czech Academy of Sciences, Prague, Czech Republic, and the University of Sheffield, UK, has identified a new protein that is fundamental to the fusion process and could open doors to novel infertility treatments. In more than half of those unable to conceive naturally, the cause of infertility is unknown. Lead investigator Harry Moore, University of Sheffield, explained, "What we know about fertility in humans has been severely limited by ethical concerns and the lack of eggs for research." End quote. In the first study of its kind, the researchers used an artificial fertilization technique to overcome these challenges. Using thousands of synthetic microbeads around the same size as a human oocyte, the researchers created artificial eggs with different peptides on their surfaces for the sperm to bind to. The beads were incubated with sperm and, using an in vitro sperm binding assay, the researchers found that not all the beads had sperm attached to them. Celia, say what? Important function of neurons' tiny hairs. Researchers have uncovered a synapse on neurons' tiny hair-like structures which may facilitate long-term changes to genomic information in the nucleus. Excuse me, that was a native ad from Neuroscience News itself, referring to another article of its own. So, yep, so it's called Celia Say What? I think I want to read that one next. However, this article on this is not Neuroscience News, this is Biotechniques.com, Biotechniques, and it's the International Journal of Life Science Methods. So Biotechniques continues. Beads with sperm attached could be identified due to the light refracting sperm heads and because they would rotate caused by sperm motility. The researchers removed beads without a sperm mate until they were only left with sperm-bound beads. Less than 1 in 1,000 beads had sperm attached. They then identified the peptide on the surface of the remaining beads and named the corresponding protein MAIA after the Greek goddess of motherhood. So I guess it's Maya. So I'll back up here, repeating. They then identified the peptide on the surface of the remaining beads and named the corresponding protein Maya after the Greek goddess of motherhood. After discovering Maya, the researchers inserted the gene coding for the protein into human culture cells. These cells responded to sperm in the same way as in natural fertilization, revealing that Maya plays a role in drawing the sperm to the egg cytoplasm and enabling the egg and sperm to fuse. Quote, the ingenious artificial fertilization technique, which enabled us to identify the Maya protein, will not only allow scientists to better understand the mechanism of human fertility, but will pave the way for novel ways to treat infertility and revolutionize the design of future contraceptives, end quote, said Moore. This discovery marks a step forward in our understanding of human fertilization. Alan Pacey, the University of Sheffield, concluded that, quote, it would have been impossible to discover Maya without the use of artificial beads to replicate the surface of human eggs, as we simply wouldn't have been able to get enough eggs to do the experiment. A classic case of thinking out of the box, end quote. Next, the researchers will explore if sperm from different individuals bind to Maya differently, as it has been theorized that some sperm may be incompatible with certain eggs. Source: Vondrakova, J. Florikova, M. Dead, D-E-D-L, and others. Maya, Fc receptor-like 3 supersedes Juno as Izumo-1 receptor during fertilization. Science, ADV, page 836. I don't know what these other designations are. EABN 0047 2022. Click here to view the press release. In keeping with this being a World Reading Club project, and this is Biotechniques Reading, Biotechniques News Reading for World Reading Club, I am going to click to read the press release. It should be a fresh press release. This was just released September 14th. So yes, this press release is from the 7th of September. Here it goes. It's from the University of Sheffield News, 7 September 2022. Scientists discover new protein which helps sperm fuse with an egg and could improve fertility treatments. This is from the University of Sheffield. Scientists have discovered a new protein which helps sperm fuse to an egg and is fundamental for human fertilization. Bullet points. New Maya protein, named after the Greek goddess of motherhood, helps to draw the sperm into the egg cytoplasm for completion of fertilization. Now, this is a beautiful image they have here. I'm going to use it to share. Infertility is unexplained in more than half of people who cannot conceive naturally. The fundamental discovery made by scientists at the University of Sheffield using pioneering artificial fertilization techniques could in future help to improve fertility treatments and help develop new contraceptives. You're listening to Biotechniques News Reading, presented by Hakeem Ali-Bocas Alexander on the Wisdom app for World Reading Club in association with UniquilibriuM. I'm preparing a quick share here to test the technology here and to promote this reading, World Reading Club. And so many tools here. It's quite interesting. I could actually copy and paste directly from in the Wisdom app while I'm broadcasting, which is nice, which allows me the freedom to much more quickly share certain information and get another handle on this and post. Right. So Biotechniques News Reading continues with scientists discover new protein which helps sperm fuse with an egg and could improve fertility treatments, from University of Sheffield News. Currently, infertility is unexplained in more than half of people who are unable to conceive naturally. In the first study of its kind, the international team of researchers led by University of Sheffield created artificial eggs using thousands of beads. Each of these beads had a different piece of protein known as a peptide on its surface so that sperm could bond with them. When sperm were incubated with the beads, scientists found only a small number of beads had sperm attached to them. After several painstaking rounds of removing any beads that didn't have sperm bound to them, researchers were eventually left with beads corresponding to one particular protein, Maya, and sperm bound to all of these beads. The gene corresponding to Maya was then inserted into human culture cells, and these became receptive to sperm in the exact way that it would during the natural fertilization process. The findings, published in Science Advances, reveal that during the process, Maya is responsible for drawing sperm into the egg for fertilization. Professor Harry Moore, lead investigator of the study from the University of Sheffield School of Biosciences, said, Infertility is unexplained in more than half of those who struggle to conceive naturally. What we know about fertility in humans has been severely limited by ethical concerns and the lack of eggs for research. The ingenious artificial fertilization technique, which enabled us to identify the Maya protein, will not only allow scientists to better understand the mechanisms of human fertility, but will pave the way for novel ways to treat infertility and revolutionize the design of future contraceptives. The findings could help to confirm the theory that some sperm may not be compatible with some eggs. Researchers now plan to explore whether sperm from different individuals bind to the protein differently. Professor Alan Pacey, co-author of the study and head of the University of Sheffield's Department of Oncology and Metabolism and Infection, Immunity and Cardiovascular Disease, said, "This discovery of the Maya protein is a major step forward in how we understand the process of human fertilization. It would have been almost impossible to discover without the use of the artificial beads to replicate the surface of human eggs, as we simply wouldn't have been able to get enough eggs to do the experiment." A classic case of thinking out of the box. The paper can be viewed in full here. Interesting. You've been listening to Biotechniques News Reading, presented by Hakeem Ali-Bocas Alexander for World Reading Club in association with UniquilibriuM on the Wisdom app. So here we have now, at the end of this article from University of Sheffield, the University of Sheffield, there it says the paper can be viewed in full here. So this is the paper that was used in this news article by the University of Sheffield. And again, in keeping with this being World Reading Club, this will now be my third cold reading in a row here of an article now referred to from Biotechniques International. And I just went down the rabbit hole here to get to these articles. And now I'm currently at the end of the University of Sheffield's article, Scientists Discover New Protein Which Helps Sperm Fuse with an Egg and Could Improve Fertility Treatments. Previous to that, I was on biotechniques.com for Biotechniques International, and it led to the article here on the University of Sheffield. And so that's where we are now. And so it will be the third reading approaching here on the Wisdom app. So once again, preparing to read this article as I post on Twitter a few images that I just captured and the information about these particular articles here, so that if anybody wants to access this live, because it's going to go for a few more moments, they can definitely join in. You've been listening to Biotechniques News Reading, presented by Hakeem Ali-Bocas Alexander on the Wisdom app for World Reading Club in association with UniquilibriuM. Continuing with, as we click here from science.org, Science Advances. Research article, Developmental Biology, Maya. Okay, here we are on Science Advances. So this is turned to a Science Advances reading. All right, currently, you're listening to Science Advances reading presented by Hakeem Ali-Bocas Alexander for World Reading Club in association with UniquilibriuM on the Wisdom app. The article here in Science Advances on science.org is titled Maya, Fc receptor like 3 supersedes Juno as Izumo-1 receptor during human fertilization, by Jana Vondrakova, Mikhaila Frolikova, and Katerina Komarskova. Science Advances, 7th of September 2022, Volume 8, Issue 36. Abstract. Gamete fusion is a critical event of mammalian fertilization. A random one-bead, one-compound combinatorial peptide library presented synthetic human egg mimics and identified a previously unidentified ligand as Fc receptor-like 3 named Maya after the mythological goddess intertwined with Juno. This immunoglobulin superfamily receptor was expressed on human oolemma and played a major role during sperm-egg adhesion and fusion. Maya forms a highly stable interaction with the known Izumo-1/Juno sperm-egg complex, permitting specific gamete fusion. The complexity of the Maya isotype may offer a cryptic sexual selection mechanism to avoid genetic incompatibility and achieve favorable fitness outcomes. You're listening to Science Advances Reading, presented by Hakeem Ali-Bocas Alexander for World Reading Club in association with UniquilibriuM on the Wisdom app. Science Advances continues with an introduction. A key event of vertebrate fertilization is the membrane fusion of the gametes, enabling the spermatozoon to enter the ooplasm and trigger the resumption of meiosis. Molecular recognition between the plasma membranes in the mouse involves the egg receptor on the sperm, Izumo-1, a member of the immunoglobulin Ig superfamily, and the sperm receptor on the egg, Juno, a glycosyl phosphatidylinosyl. Okay, hold on for a second. A glycosyl phosphatidylinositol. Good. A glycosyl phosphatidylinositol. Good. Here we go. So restart. Molecular recognition between the plasma membranes in the mouse involves the egg receptor on the sperm, Izumo-1, a member of the immunoglobulin superfamily, and the sperm receptor on the egg, Juno, a glycosyl phosphatidylinositol anchored non-functional folate receptor family member. Binding of these receptors is facilitated by a tetraspanin network, CD9 and CD81, in the egg membrane, such that deletion of either receptor or the tetraspanins substantially inhibits gamete fusion despite an apparently normal sperm-egg attachment. Juno/Izumo-1 interaction is conserved in mammals, including marsupials. However, gamete fusion displays a species-specific interaction that is particularly stringent for human fertilization. This species specificity and the observation that Juno is lost early in gamete contact before completion of membrane fusion indicate that additional membrane recognition mechanisms are likely to be involved. We sought to identify new human sperm-egg binding/fusion epitopes by using a random one-bead, one-compound combinatorial peptide library to represent synthetic egg mimics. Repeat, we sought to identify new human sperm-egg binding/fusion epitopes by using, well, maybe that's epitopes. That would make much more sense. Repeating, we sought to identify new human sperm-egg binding/fusion epitopes by using a random one-bead, one-compound OBOC combinatorial peptide library to represent synthetic egg mimics. Results. FCRL3 is identified as candidate human sperm-egg fusion epitope and co-evolved with Izumo-1. Microspheres as three-dimensional substitutes for oocytes were used in combination with a cyclic OBOC library comprising of greater than 47 million permutations, with each bead displaying a unique 6-mer random L-amino acid peptide flanked by D-cysteine residues, making them resistant to proteolysis. Library beads, around 90 micrometers in diameter, were comparable in size to human eggs, around 100 to 120 micrometers in diameter, and used in an in vitro sperm binding assay. The few beads with sperm bound were readily identified by their light refracting sperm heads and bead rotation due to sperm motility. Okay, repeating. The few beads with sperm bound, less than 1 in 1,000, were readily identified by their light refracting sperm heads and bead rotation due to sperm motility. Significantly, this sperm adhesion imitated sperm bound to oocytes in vitro with acrosome-reacted sperm pivoting about their equatorial region where sperm-egg fusion occurs. Wash beads with greater than 25 sperm attached were recovered for amino acid sequencing, 85% success rate, with the majority of identified peptides associated with cell adhesion properties. Another round of sperm binding with homogeneous bead aliquots identified peptide sequences that consistently bound sperm from all donors. Calcium is required for sperm-egg fusion, and therefore, depleting calcium from the medium can indicate whether the sperm binding assay with beads was representing a potential physiological mechanism. A particular bead aliquot, bead 16, bound 50 to 100 sperm per bead, mean 69, with calcium, but it had much lower sperm binding without calcium, mean 7, potentially indicating a post-acrosomal adhesion mechanism in keeping with gamete fusion. The sequence CAMWNEDC showed homology with the conserved immunoglobulin domain of Fc receptor-like 3, FcRL3, a membrane receptor of the Ig superfamily. Human oocytes express FcRL3 mRNA and Fc gamma receptors, closely related to FcRL3 on the oolemma. Monoclonal antibody to FcRL3 inhibited sperm adhesion to CAMWNEDC beads. Ionic calcium, while the control antibody, MAB 18.6, only inhibited sperm binding in the absence of calcium. Spermatozoa pre-incubated with monoclonal antibody to FcRL3 and Izumo-1 and incubated with zona-free hamster oocytes, used as a clinical assay of sperm fertilizing capacity, blocked both sperm attachment and fusion with zona-free hamster eggs in vitro compared with the control MAB 18.6 monoclonal antibody. An antibody against Izumo-1 also blocked sperm attachment to the CAMWNEDC beads, suggesting a relationship between Izumo-1 and the FcRL3 binding motif. Therefore, we performed evolutionary rate covariation, ERC, to analyze coevolution between proteins known to be involved in mammalian gamete fusion. ERC is a sequence analysis technique that identifies genes with correlated changes in rate across species. ERC was previously shown to find correlations between co-functional genes across diverse functional groups. We evaluated ERC-based functional relations between not only FcRL1/3 and Izumo-1, but also DCST1/2, FIMP, TMEM95, SOF1, and SPACA6 as male reproductive tissue, sperm and testes, specific proteins, except SPACAC, in which minor mRNA expression was also detected in the ovary but not oocytes. As expected, recognized fertilization genes showed highly elevated ERC values. FCRL3 also displayed significantly elevated ERC values with Izumo1, SPACA6, DCST1, TMEM95, DCST2, and Juno, comprising a co-evolving group with the top 5% of genome-wide values. FCRLs are expressed primarily on B cells and have an immune modulating function, although extracellular ligands for the receptors are unknown. FCRL3 displays an Fc homologous extracellular domain, a transmembrane domain, and cytoplasmic immunoreceptor tyrosine-based activation and inhibitory motifs, ITAM and ITIM. In contrast, FCRL1 is the only FCRL expressing two ITAM-like but no ITIM sequences and is assumed to be a co-activation receptor. Anti-FCRL1 antibody failed to inhibit sperm oocyte fusion. Therefore, our investigations focused on the FCRL3 receptor. Furthermore, TMEM71, which displays homology with the selected CAMWNEDC peptide, is not known to be expressed on gametes and, moreover, does not contain Ig-like domains necessary for fusion. You're listening to Science Advances Reading, presented by Hakeem Ali-Bocas Alexander on the Wisdom app for World Reading Club in association with UniquilibriuM. Science Advances Reading continues. FCRL3 interacts with Juno on human oolemma and was named Maya. FCLR3 protein is localized within the oolemma of unfertilized human oocytes and movie S1, and we addressed its close interaction with candidate binding proteins, Juno and CD9, with co-localization and a proximity ligation assays, PLAs. We addressed its close interaction with candidate binding proteins, Juno and CD9 with co-localization and proximity ligation assays. The strongest interaction between FCRL3 Juno reflected a high Pearson correlation coefficient. Therefore, we named the FCRL3 on human oocytes Maya, after the mythological goddess intertwined with Juno. Double immunogold labeling with gold particles showed close association between Juno and Maya along the oolemma microvilli. Maya induces sperm fusion with transfected CHO and HEK293 T cells, and paralog FCRL5 might suggest a potential role of FCRL protein family in reproductive processes. Chinese hamster ovary, CHO, cells were transfected with FCRL3 and Juno carrying plasmid and co-incubated with capacitated human sperm, 37 degrees Celsius, which adhered avidly, 15 to 30 sperm per field, control 1 per field, and displayed characteristic oscillations of the sperm head, movies S2 and S3. Double transfected, I believe that's referring to when I say movies, the videos that are here in the articles itself from Science Advances. I may edit those out in my reading moving forward. Sperm incubated with co-transfected embryonic kidney, HEK, 293 T cells displayed a range of tail beating observed previously, which can be reflected in the status of sperm cell binding as a prerequisite of fusion, with their viability remaining. And movie S5, hold on. I have to make a correction. Movie S5 is not a video or a setting here on Science Advances. It is my own misreading. And so I'll continue reading it through and research further for the understanding of exactly what that label is signifying. So you're listening to Neuroscience News, or actually Science Advances reading presented by Hakeem Ali-Bocas Alexander on the Wisdom app for World Reading Club in association with UniquilibriuM. This is getting interesting. So Science Advances continues. Sperm incubated with co-transfected human embryonic kidney, HEK, 293 T cells displayed a range of tail beating observed previously, which can be reflected in the status of sperm cell binding as a prerequisite of fusion, with their viability remaining. And movie S5. Using artificial intelligence, AI, object identification, we developed novel neuronal network training masks for human sperm tail beating pattern quantification. On the basis of the AI findings, the difference between HEK cells or HEK cells, non-transfected, transfected with Maya or co-transfected with Maya and Juno, was significant in the amount of sperm displaying low and medium amplitude of tail beating. In particular, 75% of sperm incubated with co-transfected cells and showed medium amplitude beating pattern, which we interpret as a pattern of sperm binding and the most favorable for future possible fusion. We emphasize that our system took into account the data distribution between individual human sperm tail movement amplitudes, reflecting those observed in green fluorescent protein GFP fusion assay. The majority, P is less than 1,000th of these sperm, later stop with a hardly noticeable movement or straighten flagellum, a behavior reminiscent of the moment of sperm-egg fusion in vitro. And movies S5 to S7. Previously, it was shown that cells expressing Juno alone bind spermatozoa but do not fuse with them. Therefore, to monitor the trajectory of this sperm cell entry, HEK293 T cells were co-transfected with Maya, FCRL3, Juno, and plasmid encoding with a GFP tag farnesylated KRAS to visualize the cell membrane. And movie S8. First, we detected significantly elevated sperm binding to these co-transfected HEK cells. And second, we verified the ability of sperm to fuse with these cells as visualized by segmentation and volume rendering of fluorescence using Imaris 3D and movie S9. Notably, when the human sperm were protein were pre-incubated with the synthetic CAMW-NED peptide, sperm binding to HEC293 T cells decreased, indicating a possible interference with the Ig domain interaction. Third, we combined correlative light and electron microscopy, CLEM, to target the site of sperm binding to the co-transfected cell with focus ion beam scanning electron microscopy, FIB scanning electron microscopy, volumetric imaging, and found the sperm head to be exposed to the cytoplasm of this co-transfected cell by FIB scanning electron microscopy, and I2 and movie S10. Yeah, so these are referring to panels here in the article. So I will self-edit live here as I'm reading from now forward, unless I feel like it's necessary to read those designations to avoid confusing myself. Science Advances reading continues. The reconstruction of FIB scanning electron microscopy dataset showed membrane fusion over the equatorial region of the sperm head extending back over the post-acrosomal region as at the beginning of sperm-egg fusion. Human sperm will fuse with lymphocytes, which also highly express FcRl3, and inhibition of FcRl3 gene expression by FcRl3 short interfering RNA, siRNA, led to decreased sperm fusion with GECO-1b lymphocytes. Furthermore, a soluble extracellular domain of FcRl3 protein bound to Izumo-1, which was relocated to the equatorial segment after the acrosome reaction. You're listening to Science Advances Reading, presented by Hakeem Ali-Bocas Alexander for World Reading Club in association with UniquilibriuM here on the Wisdom app. Science Advances Reading continues. We confirmed the expression of transfected proteins, Maya and Juno, along with the cis protein interaction between Maya and Juno within the cell membrane, and the transprotein interaction between Maya and Izumo on the sperm. Izumo-1 protein in human sperm was used as a control. PLA performed on Juno Maya double transfected cells with bound sperm revealed an interaction between Maya and Izumo proteins, and indicating a reciprocal binding activity of these two proteins with respect to controls. A sperm cell GFP, that's green fluorescent protein fusion, assay was designed, and human sperm transfected with GFP plasmid were added to Maya or Maya and Juno co-transfected HEC cells. When sperm cell fusion occurred, the GFP signal expressed by the cells was detectable. The sperm fusion with cells co-transfected with Maya and Juno was significant. P is less than 0.5, compared to the single transfected Maya group, supporting our proposal that Maya facilitates sperm fusion in the presence of Juno. Transfected cells fused with sperm still had a detectable sperm tail outside the cell, similar to sperm oocyte fusion. Okay. Now I see what I have done here is to perhaps bite off a little more than wisdom would like for me to chew, or at least me on wisdom. So I'm looking at the progress bar here on this article from science.org, which is the home of Science Advances. And there is a lot to go in this very heavily technical article that I don't mind reading, but in some ways I do, because I want to move on to other projects. So in keeping with that, I'm going to go back to the top here. So this is a Science Advances article. Science Advances article is on science.org, and it's called Maya, or MAIA, FC receptor like 3 supersedes Juno as a Zumo-1 receptor during human fertilization. And so what they're talking about here is that this new receptor is doing better than a previous one that they were using for this other Zumo-1. So Zumo-1 is now reported in this article to be a better receptor, or I'm sorry, to be received better to Maya than Juno during human fertilization. So this is what they're discovering, that that Maya is a better receptor site than Juno. So I suppose that's a better way to sum it up. And then I'll conclude with their abstract once again. So the abstract on Science Advances of Maya FC receptor like 3 supersedes Juno as a Zumo-1 receptor during human fertilization. Gamete fusion is a critical event of human, gamete fusion is a critical event of mammalian fertilization. A random one bead, one compound combinatorial peptide library represented synthetic human egg mimics and identified a previously unidentified ligand as FC receptor like 3, named Maya after the mythological goddess intertwined with Juno. This immunoglobulin superfamily receptor was expressed on human oolemma and played a major role during sperm-egg adhesion and fusion. Maya forms a highly stable interaction with the known Izumo-1 Juno sperm-egg complex, permitting specific gamete fusion. The complexity of the Maya isotype may offer a cryptic sexual selection mechanism to avoid genetic incompatibility and achieve favorable fitness outcomes. You've been listening to a combination of Biotechniques News Reading, University of Sheffield News Reading, and Science Advances News Reading, presented by Hakeem Ali-Bocas Alexander on the Wisdom app for World Reading Club in association with UniquilibriuM.